Hello and welcome to the Genome Sequencing COVID-19 webinar. My name is Dr. Hayley Teasdale and I'm from the Australian Academy of Science. This is the third event in a series focusing on international scientific collaboration in the fight against the COVID-19 pandemic. Firstly, I would like to acknowledge the traditional owners of the land on which we meet today. For me, it's the Ngunnawal people. I would like to pay my respects to their elders past and present. Genome sequencing has been a vital tool during the pandemic to understand the biology and evolution of COVID-19. It's also helped shape the public health response, with authorities using the information to provide timely and critical clues to track clusters and identify how infections are linked. And here to discuss the intricacies of genome sequencing are Professor Ben Howden from the Doherty Institute in Melbourne, Australia. He's the Director of the Microbiological Diagnostic Unit Public Health Laboratory and Co-Chair of Australia's Communicable Diseases Genomics Network. We also have Dr. Tereza de Vasconcelos from the National Laboratory of Scientific Computing in Brazil, and Dr. Ming-Kun, Principal Investigator of the Beijing Institute of Genomics at the Chinese Academy of Sciences, and Dr. Alessandro Marcello from the International Centre for Genetic Engineering and Biotechnology in Italy. Thanks to you all for joining us for this discussion. Before we ask our panellists some questions and get the conversation started, we have some messages from Brazil, China, and Italy. Please welcome His Excellency Tim Kane, Australian Ambassador to Brazil.
As Australia's Ambassador to Brazil, I warmly welcome this initiative of the Australian Department of Industry, Science, Energy and Resources and the Australian Academy of Science on the genome sequencing of COVID-19. Australia and Brazil share a close and growing relationship. Brazilians are now the largest Latin American community in Australia. Brazil is one of our largest trading partners in the region and an important investment destination. A shared challenge we face is the pandemic, and we need to continue to work together to overcome it. Science and research cooperation is an increasingly important part of our relationship. In 2017, Australia and Brazil signed a Science, Technology and Innovation Agreement. Brazil is Australia's largest research partner in Latin America and our 15th largest globally. Between 2017 and 2020, Australian and Brazilian scientists produced over 8,000 joint publications. We aim to enhance our cooperation, including through the Australia-Brazil Research Collaboration Week, which we will host this year with the Australian Academy of Science. I welcome the participation of Dr. Tereza Ribeiro de Vasconcelos of the Brazilian National Scientific Computing Laboratory in this event. Dr. Ribeiro de Vasconcelos' significant expertise in DNA sequencing is at the forefront of COVID-19 research and is making a considerable contribution to the global fight against COVID-19. I thank everyone for the important work you are doing and extend my best wishes for this event. Thank you.
Thank you, Ambassador Kane. Now please welcome His Excellency Graham Fletcher, Australian Ambassador to the People's Republic of China.
Good afternoon from Beijing. I'm pleased to be here today for the Australian Academy of Sciences' third COVID-19 webinar, which will bring together experts from Australia, China and Italy to share their experiences and lessons learned during the COVID-19 pandemic. Australia and China have a well-developed science and research relationship which has grown over more than 50 years. This relationship is underpinned by a Science and Technology Treaty, with last year marking its 40th anniversary. By collaborating and taking advantage of our strong science and research capabilities, we've been able to address a range of scientific challenges that have benefited both our countries and the international community more broadly. Collaboration on COVID-19 response is another noteworthy example, It's been a tough 12 plus months for many countries around the world, including Australia and China, as we've responded to COVID-19, and I'd like to take the opportunity to acknowledge the hard work done here in responding to the pandemic. I have no doubt the ability for countries to respond effectively would have been made more difficult but for the significant science and research work that has occurred in recent years, including on such matters as genome sequencing, which has been crucial to identifying chains of transmission. I wish you all the best for your discussions and hope that they provide valuable information and connections that will assist you to continue your important COVID-19 work.
Thank you, Ambassador Fletcher. Now please welcome Her Excellency Margaret Twomey, Australian Ambassador to Italy.
Hello, my name's Margaret Twomey and I'm Australia's Ambassador to Italy. Life has not been the same here since Italy identified its first case of COVID-19 on 21 February 2020 and went into lockdown. Now, a year later, Italy is confronting new variants of the COVID-19 virus and has recently passed the sobering milestone of 100,000 deaths. This webinar is a timely opportunity to recognise Italy's leading role in the pandemic as the first Western country affected and the first to apply science to contain it. In particular, I would note Italy's role in genome testing and its early work in sequencing variants, and its role in the development of the Oxford-AstraZeneca vaccine. During my brief time here as Ambassador, lockdown though it has been, I've been struck by Italy's important role in hosting international science bodies and by the strength of scientific cooperation between Australia and Italy in fields such as space exploration and radio astronomy. Our recently signed bilateral science treaty will help expand such cooperation. For my own part, I look forward to working with Italy during its host year of the G20 in providing the political support for the science that will end the pandemic globally. Thank you and best wishes from Rome.
Thank you, Ambassador Twomey. We appreciate these messages and the positive collaboration between our countries to fight this pandemic. So now to our panellists. Let's start with you, Ben. Could you tell us what exactly is genome sequencing and how has it been used prior to COVID-19?
Thanks very much, Hayley. Well, genome sequencing is a process for revealing the whole genetic code of a species. And in this case, we're talking about a pathogen that causes human disease. And so that process really uncovers every aspect of the genetic code of the pathogen. And each of the pathogens that we work on has a different size genome, and the genome size for the SARS-CoV-2 virus that causes COVID-19 is about 30,000 bases. And so the process that we undertake to reveal the genetic code is to take the samples that have been used for diagnostic testing. We take them into the laboratory, extract nucleic acids from those samples, and then we go through a wet lab process to prepare a library that goes onto a genome sequencing machine. After that happens, what we get is a file full of short or sometimes longer reads, which are really strands of letters which come together to make up the genome of that virus. And then we go through the really important steps of bringing them together using bioinformatic processes to compare the genomes out of each sample to others and to draw conclusions about the type of virus and the mutations that might be present in it. Before COVID-19 started, pathogen genome sequencing has really been rapidly increasing over the last 20 years. It really started as a research tool where we used it to understand how pathogens, in particular bacteria, but also other types of pathogens, cause disease, how they spread, how they might become resistant to treatments, and how we might respond to them by developing vaccines. But over the last decade, and particularly in the last 5 years, genome sequencing has really become a critical public health tool. That means it's moved into public health laboratories who work on a day-to-day basis with public health units and analyse genome data of pathogens to inform public health responses.
Sure. Could you perhaps give us an example of one of the other pathogens that genome sequencing has been a really powerful tool in combating, perhaps?
Yeah, so on a global scale at the public health interface, genome sequencing has really been heavily used for food-borne diseases, so things like listeria, and Salmonella, where contaminated food can cause outbreaks across countries or even across the world. And using genome data, which is very interchangeable, and through sharing that data, much greater accuracy and earlier detection of outbreaks using that process.
Oh, that's fascinating. And was there any part of this technology that was particularly adapted for the fight against COVID-19? Or is there any previous technologies that were used? Could you tell us about that?
I think at the public health interface, which is where I sit in a public health laboratory, what we've done over the last 5 years or so is work out how to bring this exciting technology of genome sequencing from the research lab into the public health lab. And that means we've had to do things like get our labs kitted up with the right equipment to perform genome sequencing. We've had to bring bioinformaticians who previously mainly worked in the research environment into the public health labs and start working in more of a diagnostic public health environment. And we've started to have to get our processes ISO accredited, so we're really working at that high standard of reproducibility and accuracy. And I think by doing that with bacterial diseases such as Salmonella and Listeria over the last 5 or 10 years, we were ready to apply this technology at scale when the COVID pandemic started.
Wonderful, thank you. Now let's hear from Anna Tereza. Perhaps, Anna Tereza, you could tell us, what does genome sequencing tell us about how SARS-CoV-2 evolved?
So, good morning, thank you for inviting me. So the SARS-CoV-2 genomes, as my colleagues said before me, genomes encodes instructions that are organized into sections called genes to build the virus. So the genome sequence decodes the genes, allowing us to monitoring how the change occurs over the time. Virus evolves by mutating. That is, there are changes in their genome codes. Understand how these changes affect the characteristics of the virus permits to use this information to predict how it might impact the public health.
Interesting. So on this topic, Dr. Anthony Ashton has asked us via email, did SARS-CoV-2 jump once from a single animal, long since eaten, or multiple times from multiple animals?
So the question is the animal that infected the first person. So far as we know, bats appears to be the first suspect, because when we look at the phylogenetic tree, we see that the bat coronavirus is the closest relative to the SARS-CoV-2, sharing about 96% of their genomes. But there is another animal, a small mammal, called pangolin, that can be implicated as well, because the spike proteins in SARS-CoV-2 is similar to the pangolin coronavirus spike proteins. So probably the virus came from bat and passed before to arrive in human at the pangolin genome.
Well, that is fascinating. So let's move on to Ming-Kun. How has the field of genomics developed or changed in recent years?
As Ben already mentioned, the field of genomics developed rapidly after the completion of Human Genome Project in 2003, mostly benefit from the techniques and devices developed by the Human Genome Project. And in 2005, there are so-called next-generation sequencing platform emerged. And the first-generation sequencer only sequenced single DNA molecules at a time, while the next-generation sequencing platform could sequence millions of fragments simultaneously. And the third-generation sequencing platform emerged in 2011, which featured by ultra-long sequencing read lengths. And moreover, scientists has developed powerful tools to analyze the function of the genome, such as CRISPR gene editing technology. And scientists have identified many functional genes and non-coding elements that regulate the development and diseases. And based on that, scientists have developed a molecular targeted therapy and gene-based personalized medicine, which is supposed to be more efficient in treating diseases. Today, we are talking about viral genomics, and during SARS pandemic in 2003, it took more than 3 months to get the genome of the pathogen. And with the development of the new technology and methodology, the genome of SARS-CoV-2 was obtained just in 5 days, which is really a big progress.
That's very fast.
Yeah.
And so with these challenges, with these developments, what would you say are some of the challenges and trade-offs that come along with them?
Yeah, sure. First, we still do not have a perfect sequencer. An ideal sequencing platform should have a high throughput, fast speed, high quality, long read length, and robust in different conditions. So far, there's no single machine that can meet all these requirements. And even though we do not have a perfect sequencer, we already generated massive data that cannot beyond our capability of data mining. We are still far from understanding all these genetic codes. And on the one hand, we need more data, especially phenotypic and functional data. And on the other hand, we need new algorithms to analyze the data more efficiently. Meanwhile, genome editing technologies are still immature. And besides the technical challenges, we still had some ethics issues to solve. And considering the challenging viral genomics, we still do not have good reference database, and only a small proportion of huge diversity of the virus and pathogens has been sequenced, so which lead to false negative in screening for the pathogens. And experimentally, it's still tricky to get the complete pathogen genome from clinical samples due to the limited copy number of the pathogen in the sample and the severe contamination from human genome.
So when you sequenced the genome of SARS-CoV-2 in China early in the pandemic, what did you and your colleagues find out?
Yes, we did some work on that. So far, there are in total around 2,000 complete SARS-CoV-2 genome sequenced, obtained from samples from tens of provinces in China. And around 500 sequences were obtained from samples collected before February 2020, which accounts for 4% of all confirmed cases at that time. And 2 major clusters was observed, which was called lineage A and B. And there were only 2 mutations between these 2 clusters, suggesting one was recently descendant from the other, although it was still debating which is ancestor. We have tried our best to collect the on-site date from the publications and from the submitter of the data. We finally confirmed the 13 sequences with on-site date in December 2019, which is really the earliest cases. And most of the sequences had the exactly same genome. And the other sequences had 1 to 3 mutations compared to these identical sequences. So it's a strong signal that it's not been a very long time since the virus appeared in Wuhan at that time. So some statistical analysis estimate that the ancestor of these 13 earliest samples was between mid-November and early December, which is consistent with epidemiological data.
Right, and perhaps you could explain for our audience, what do you mean when you say cluster? Could you define that for us?
A cluster is a bunch of sequences that share the same mutation.
That's well said. Excellent. So let's hear from Alessandro now. So COVID-19 hit Italy quite early on. So alongside the information devised from sequencing the virus, how important is it to have good epidemiological and clinical data alongside this?
Yes, of course, Italy was the first country in Europe to be hit by the epidemic early in February last year. And what happened is that in January, we had a couple of cases from tourists from China that were isolated in Rome, and sequences were performed and showed to be belonging to the China virus. Then this gave us kind of sense of false security. And when we had the epidemic hitting hard in northern Italy, our first question was to understand the origin of this virus circulating in Lombardy. And we couldn't really track the original case by classical epidemiological tracing. And sequencing gave us some insight on the fact that the circulating virus at the time was already circulating in Europe, probably already circulating in Germany, and already a variant, so-called variant, with respect to the original virus from Wuhan. Then, of course, we were overwhelmed by the epidemic. We had other priorities, including diagnostics, tests, and tracing, and so on. And sequencing became a little bit off the table of the priorities, and that was a bit unfortunate because Italy, if you compare the amount of sequencing performed in Italy and other countries in Europe during 2020, compared to UK, for example, there is a real gap in depth of sequencing and available sequences from these countries. Only now, after the circulation of new variants, the sequencing effort became more structured in Italy as well, and more, let's say, distributed on the territory. In 2020, only a few centers kept on sequencing. This allowed us to understand how the virus evolved in the country. So we have an idea of what happened during the summer. We had clusters of virus that then would ignite the epidemic in the second phase in September, October.
Okay, so how can we use the genome sequence to create diagnostic tests for COVID-19?
Okay, so the effort of our Chinese colleagues has been tremendous at the very early stage of the epidemic. It gave us the sequence of the, let's call the Wuhan virus, so the one that originated the pandemic. And this allowed us to have quickly a molecular test, which is still currently used for diagnosing the infection. Of course, the molecular test is based on specifically identifying the nucleic acid of the virus. So every changes in the regions that are recognized by the primers of the PCR, which is the test used for the assay, would affect the assay. And this became evident in the last months when the UK, so-called UK variant started circulating, and we had a drop-off of one of the targets of some of the assays that were used in diagnostics. So understanding the evolution of the sequence has an immediate impact on the diagnostic assay, in this case, all the molecular diagnostic assays. This has also an impact on other assays because also serology assays are based on protein of the virus that are used to recognize the immune response. And of course, any changes to these proteins during the epidemic may change the ability to identify the immune response to the virus, as well as rapid antigenic tests also rely on specific antibodies to specific region of the virus. And also in that case, mutation may affect the assay. So there's also a direct impact on other kind of assays of knowing the sequence of the virus. And of course, there is an impact on vaccine development because vaccines are based on fragments of the virus that would trigger an immune response. And of course, if the virus changes during the epidemic, also these fragments change and may be introduced in a new, let's say, variant of the vaccine.
Right, so the more we learn about the virus, the better we can detect it in our bodies and help produce vaccines against it. Is that right?
That's correct. I mean, the vaccines are successful. The one that are rolling out now are certainly protecting. Of course, we have to keep our eyes open for future. We know from the influenza that every year there are changes in the vaccine composition. This may be the case also in the coronavirus case.
With each nation finding out data separately, sharing this information and collaborating across the globe is a vital element of the pandemic response. Ben, in Australia, you were involved in sequencing cases from the state of Victoria, where the majority of Australia's cases occurred in 2020. How did you select which samples to use?
It's a great question because there was no rule book around that early on. And I guess what we did was we borrowed from our experience in working with other pathogens of public health significance. And the more you sequence, the more insights you get. And also, if you take if you take the approach of sequencing all comers, then you reduce the bias that you might have in some of the interpretations or conclusions that you drew and draw. So early on, we decided to attempt to sequence all the samples that we could gain access to from the state of Victoria. And I will say that similar approaches have been taken around the country in Australia. And I think there's been a huge benefit to that. I mean, we do have to acknowledge that on a global scale, the case numbers have been relatively low in Australia. It's allowed us to continue to pursue that strategy. But nonetheless, I think sequencing all available samples has been really valuable, and also shown the importance of, as Alessandro was mentioning, that idea of having approaches around the country and around the jurisdiction where samples are available for sequencing. There's cooperation between laboratories that are making the diagnostics and diagnosing the cases, and the public health laboratories that undertake the sequencing. That's been a really important part of this response, is to get that collaboration occurring through all of our laboratory networks, whether they be genomic sequencing laboratories or not. And so in the end, to date, we've received over 75% of all positive samples from our jurisdiction and sequenced successfully over 76% of all of those. And so it's been a very comprehensive approach. And this has really allowed us to be very certain about some of the conclusions that we've drawn from the data that we've been analysing, and to be certain around some of the implications of that data.
Right. So how do these findings from genome sequencing assist with things like contact tracing of the virus and other public health actions?
Yeah, so that's sort of exactly where the laboratory that I work in sits. And, I would say that now in Australia and in Victoria, the genome sequencing of SARS-CoV-2 has really become a critical component of the public health response. I guess the main, what Alessandro alluded to this, the main reason to do this is to find genomic clusters and then try and link them with the epidemiological data, so that we're trying to either refute or prove epidemiological ideas around how transmission is occurring. And just to give some, clear examples, in Victoria, we had an initial wave of infections that were all importations from overseas when returning travellers rapidly came back to Australia at the start of the pandemic. And by sequencing almost all of the cases, we were able to identify a very large number of genomic clusters that represented returning travellers from many countries around the world, with very little onward transmission in the Australian community, because these clusters were generally quite small. At the end of that period, once we had a very robust quarantine program in place, we were able to show that all of those early clusters had actually been eradicated from Victoria. And so that was a very powerful message for the public health units to be able to say, look, we've put these public health responses in place, we were able to eradicate community transmission. And that powerful message could then be used later when we had a second wave of infections and we needed to go back to using those public health measures. And during the second wave, some of your viewers may be well aware that Victoria had a huge second wave of infections in the second half of last year, with many hundreds of cases per day during that period. And using genome sequencing, we were able to identify that almost all of those cases came from a single breach of our quarantine practices in Victoria, from one family who'd returned from overseas. And so that really demonstrates the power of this. There would have been no capacity to really determine the source of all of these cases that were rapidly emerging in the community. And that ability to pinpoint it to a single quarantine breach has been a very important aspect of the public health response. And then finally now, I guess we're in a wave where we have very low case numbers, but we do have cases arising in hotel quarantine. And there's a real desire from our public health colleagues to have very rapid answers around the source of these new cases. Could they have been acquired in the hotel? Do they look like they've been acquired overseas or not? And then the other things we're doing, which has been very helpful with the public health responses, is looking at hospital-acquired infections in staff and patients, and also now identifying variants of concern. And so for every new case in Australia with the very low case numbers, any variant of concern case gets treated a bit differently in the quarantine process. And so that ability to rapidly identify them has become really important.
Of course. Now, how is your team building networks with other nations in the Pacific region to collaborate and learn from each other?
Yeah, it's a great question. I think what we've learned through the use of this technology at the public health interface it's a technology that needs to be accessible to all countries trying to deal with this problem. And the scale at which they'll need that will be different. But we've certainly had a lot of communication with Pacific Island nations in our region, and thankfully they've mostly not been heavily impacted by the pandemic to date. But they have found it very useful to be able to show that a small cluster of cases are all genomically linked to each other, such that they can reassure themselves that their quarantine processes are working and they haven't had multiple quarantine breaches. And so even a small number of cases from a Pacific Island country can be very usefully sequenced with that knowledge that they can take that back to consider how they continue to do their quarantine. And I think what we're seeing now is the evolution of a network of public health laboratories around our region in Australia who are very interested in understanding how to access and deploy this technology more broadly. And I think there's certainly some opportunities to do that either through efficient referral and networking approaches where we support our partners, or in future even deploying the technology into some countries to act as additional nodes of capacity.
Now, Tereza, by studying the genomics of the virus in Brazil, you discovered a particular new variant. How did you find out about the P2 variant?
So since the beginning of the pandemic in 2020, we start to sequence in Brazil, collaborating with different groups from the university and research institute here in Brazil. At the beginning, we sequenced and we characterized that in Brazil, we had two main lineages, the B1128 and the B1133. Past some months, in the beginning of October, since the end of November, we sequenced another round of genomes. The genomes came from the Rio de Janeiro state in Brazil. And at this time, we realized that the mutation that the name is A484K appears in the lineage that was before named B1128. At this time, this lineage sequenced here in the Rio, and after we sequenced in another region in Brazil in the south, in Rio Grande do Sul, and now last week we finished from the northeast of the country, and this new lineage P2 is spread all over the country, starting Rio probably in July, according to the phylogenetic tree, and at this day he is spread all over the country. This mutation, the name is the escape mutation, is very important because it reduces the neutralization activity in convalescent plasma of COVID-19 patients. So, as we call the variant of interest for the community.
Right, now they give those variants fun names, don't they? They're not very catchy. Now, did you need to corroborate and compare this with other variants? I'm interested, why is strain surveillance important internationally?
Yes, we have all the time to compare the genomes with the first genome from the Wuhan, but also with the genome around the world. We have to compare because the virus always mutating. So we have to verify if there is any mutation that enter in some protein, especially in the protein, the spike protein is very important. They can interfere in the vaccine or in the test. So the genomic surveillance is very important all the time. At this day that we can travel, we can commute the genomes, all the time. So it's very important to perform quality epidemiology genomics.
Can you tell us how you use supercomputers to assist in your work?
So we have sequenced a lot of genomes around the world, and the scientists is deposited in the different database, and we have to develop some pipelines to compare, to assemble the genome, to extract information from the virus. So at the lab, you use the supercomputer to extract this information, to organize the data, and to use some techniques for machine learning, for example, to identify some patterns that is important from a specific lineage, or to identify some clusters that can appear in the virus. But one more important thing that we are now have to study also not only the virus, but the patients too. So we have to perform the sequence from the host to the human. So we have to do we are doing some experiments, some transcriptome analysis, some exome analysis, some microRNA analysis, and to analyze faster this information is easier to have a supercomputer that can use some techniques from HPC to interpret this data.
Of course. Okay. So, Ming-Kun, can you tell us an example of a collaboration, perhaps from China or from your institute, that has assisted in the fight against COVID-19?
Yes, collaboration is very important in fighting against the COVID-19. As a computational biologist, I had many collaborations with clinicians, biologists, and molecular biologists. And our institute was involved in the investigation of the sourcing of local outbreak to identify the possible source and transmission chain of virus. We have a very big consortia consistent of different departments and institutions. And once there is a new infected cases, we had the regional CDC to collect the specimens from the patient and environment and conduct the epidemiology investigation. We have another group to extract the nucleic acids and doing molecular tests to screen for SARS-CoV-2 positive samples. Another two groups in charge of laboratory preparation and sequencing. Once the data was generated, at least two groups will assemble the viral genome and identify its closest relatives independently. So this process normally take 48 hours. Then we will work together to go through all the evidence to find out all possibilities and return the result to the authority. I think such collaboration is a very efficient way in dealing with emergencies.
Right, so how can genome sequencing help us to expose infection control failures and trace these small sporadic outbreaks?
Yes, the situation in China is very similar to what Ben has described in Australia. So let me take the outbreak investigation in Beijing in June last year as an example to show how genome sequencing can help us to trace small local outbreak and develop new prevention and control policy. So in total, over 300 people got infected in that outbreak, from which we got 70 high-quality viral genomes. And all the genome sequences shared the same 8 mutations. Over half of the genome had no other mutations. So most of the rest of the sequence had only one mutation. This finding suggests that there was only one ancestral virus strain introduced in this outbreak. And the introduction time was estimated to be at the end of May. The genome sequence is different from the viruses in preceding outbreaks in China. So this indicates that the strain was unlikely to be derived from strains previously circulating in China. And we then sequenced all the imported goods that sold in the store, which were suspected to be the origin of the outbreak. And in the surface of the salmon fish, we got a partial viral genome that identical to the strain circulating in the patients. So this evidence suggests that the contaminated salmon was likely to be the source of the outbreak. And the authorities then strengthened the surveillance of the product imported from the pandemic regions.
Right, now we've touched on it a little bit earlier, but I was wondering if you could tell us how genomic sequencing informs the production of vaccines. How does that work?
Yeah. So first, there are different types of the vaccines. Genome sequencing is essential for some of them, like the genetic vaccines, the viral vector vaccines. For these types of vaccines, we need to identify the genetic code, namely a gene or a subset of a gene whose products can be recognized by the immune system and stimulate an immune response. So after identify these genome segments, we then need to deliver them into our body through viral vector or lipid nanoparticles. After that, we can also design pseudoviruses by inserting the antigen sequences of SARS-CoV-2 into non-replicable vectors to evaluate the efficacy of the vaccines in a lab with lower biosafety level to accelerate the vaccine research and development. Besides that, we know RNA virus mutates very fast. Some mutations on the virus genome may help them to escape the antibodies triggered by the vaccines. So we need to do genome surveillance to look for possible escaping mutations. And once the variant became dominant in the population, we may need to update the vaccine by incorporating the new antigen sequences in the vaccine.
Right. Now, Alessandro, have there been any barriers to international collaboration?
Well, I must say that the international efforts have been tremendous. Communication in science has been unprecedented with different tools from previously. Preprints, so meaning papers coming out before peer review, social media being used heavily between researchers. That's been really helpful in circulating essential information to tackle the epidemic. Clearly, barriers still exist, not really in the communication between scientists or in the availability of sequences, for example, on public repository that has been very efficient, is the availability of sequencing from certain countries that cannot afford sequencing or have difficulties in these techniques. As an international organization, we made an effort to make sequencing available to our constituency. So we had we helped sequencing countries like Moldova, Iraq, Santo Domingo, and several African countries. And they couldn't afford sequencing. We made this available to them, and so we had an idea of what was circulating in the country. And this is important. It's very important because the virus now is globally spread, is circulating in different countries, and variants of concern are emerging from different places, not only from Brazil or UK or South Africa, but we have other possible variants coming from different countries. And this we sequence more spread across the globe, we would have a delay in understanding if a variant is emerging. And this is particularly relevant, for example, in Africa, where we already had the South African variant of concern being detected, and the Nigerian one as well. And there could be a reservoir of potentially harmful variants.
Right. Now, we've been asked by a few people online about the use of individuals' genetic material. So where did you source your positive SARS-CoV-2 samples from? And what happens to a person's genetic material once it's been tested or sequenced?
Okay, that's an important issue related to bioethics. I mean, we had put in place a framework to archive samples from patients or individuals being tested and sequenced that allowed us to work in a ethical framework in with our samples. Here there are two issues here. One is the sequence of the virus and the association of the sequence of the virus with a specific patient or individual. Other thing is the genetic material of the individual. This is also important because we have to understand what are the variabilities in the response to the virus by different individuals, so by the different genetic background. And this is covered by ethical agreements and decision made by the ethical committees. Clearly, I must say that this epidemic has also showed that there's been an incredible acceleration of the need of these ethical frameworks to support research. And we cannot wait to have, for example, informed consent from all the patients to really start doing the work. So this has to be somehow solved. I think there is a requirement to have better and more easy access to samples to be able to have a fast response to an ongoing epidemic. So on one side, we have now a day-by-day understanding of the evolution of the virus. On the other hand, we need to have the possibility of studying it in real time. So I don't know if it's clear, but it's clear there is a framework. The bioethic is an issue. We need to accelerate this framework.
Right, thank you very much. So we're now going to move on to some questions that have been sent in online. To start with, Anna Tereza, we have a question from both Professor Linda Blackall and Jacob Vu. They'd like to know, how do you see the future of genome sequencing for predicting the next pandemic?
Yeah, you have to implement a really all over the world a really genomic epidemiology to have in the place some place south, like in Brazil, South Africa, Africa in general, to perform all the year amount of sequence for sample for different source, and to perform a permanently epidemiology sequencing different samples from patients, from animals, to prevent how many viruses will arrive, because they will arrive all the time. It's natural, it's ecology, it's normal to do that. So surveillance, epidemiological surveillance is very important, and I think that it will be an effort for all the world to do that at the same time.
Now, our next question, I believe it would be best answered by Alessandro. It comes in from Brunio, and he says, which parts of the spike molecules are most variable or able to mutate?
Okay, there has been a great effort to map the mutations in the spike protein and the significance of these mutations with respect to recognition of the receptor or neutralization from the immune response. Clearly, now we have the structure of the spike protein. We know what are the areas of the spike protein that recognize the receptor, at least the ACE2 receptor. There are mutations that cluster in that region, which is called the receptor binding domain. However, there are also other areas of the protein that are important for the infectivity of the virus. Sequencing is great, but then it's only a first step. Then we need to understand what is the significance of the mutation in terms of ability of the virus to infect more or less, to be more or less pathogenic. And all this comes as with what's called wet lab experiments, something that we do in the lab and from epidemiological data. So, I mean, sequence is the first information. We need to understand, actually it's redundant. We have a lot of information from sequencing. We have to understand which are the ones which are more relevant, and then study those mutations in terms of the effect on the pathogenicity of the virus.
Excellent. Now, Ming-Kun, Marta has asked, what instruments did you use to isolate this exact virus?
Actually, we don't do the isolation. We just doing PCR or hybridization capture, to enrich the genetic material of SARS-CoV-2 from the clinical samples, and then directly sequence all the genetic material we got. And once we have the data, we're trying to assemble all these rates into longer contexts. And some of these contexts would be derived from SARS-CoV-2. Then we know we got the genome of the SARS-CoV-2, but we do not need to isolate the virus.
Now let's throw to Ben for a question. Rebecca Webb has asked, how are the variants being traced and reported throughout the world? And is there a bit of a time lapse between the variants being discovered and the reporting?
It's a great question, and it's a complex one because the variants are being detected through the sequencing efforts, but the epidemiological or the pathogenic impact of those variants is not necessarily known when they're first detected and predicted to be of potential relevance. And so I think there's a lot of work to link up these emerging variants, which ones truly have evidence for changes in viral behaviour. And some of that is to do with epidemiological factors, how rapidly they spread, possibly how severe the diseases they cause, and also those things that Alessandro was mentioning around laboratory experiments to look at how well those viruses bind to cells, how well they invade, and those sort of factors. And so I think we are in a place where we are trying to look to get some standardisation around the detection and reporting of these, even within Australia and our region, but also globally. We really need an effort to have a consistent approach to this. And every time a new potential variant emerges, there's going to be some concerns around that, but we need a very systematic approach to how we classify them as true variants of concern and what we do about that at a public health level within each country, depending on their current context.
Thank you, Ben. Now that's all we have time for this evening. I'd like to thank everyone for joining us, and thank you to the Department of Industry, Science, Energy and Resources. And a big thank you to our panellists, Professor Ben Howden, Dr. Alessandro Marcello, Dr. Ming-Kun and Dr. Tereza de Vasconcelos. Thanks to you all for joining us for this discussion. Our next webinar in this series will be on Tuesday the 20th of April, focused on COVID-19 and Indigenous populations. Be sure to check out the Australian Academy of Science website for all the details. And don't forget to follow us on social media for more trustworthy and fascinating science. I'm Hayley Teasdale. See you next time.